Our presentation is over the development of standardized written and video protocols for Aplysia californica research. My name is Makaela Hampton, and I'm a senior biological sciences major.
My name is Baylee Arnold, and I'm a junior biological science major.
I'm Maddie Tegeler, and I'm a junior biological science major, and then our mentor is Dr. Kimberly Hale.
The project goals for our research were to standardize protocols in order to achieve three things. The first being to enable new and returning students to contribute more to research to yield more successful results. Secondly, to perform more ethical research on the Aplysia californica, and lastly, to allow for more consistent and cohesive research. By increasing our organization and responsibility, we have been able to achieve these goals and to yield more successful results as an upper-level research course.
Throughout our research, we've needed a couple tools to keep our group organized. We have really utilized our Moodle page where every member of our research group has access to. On this Moodle page, we keep our written protocols that every member of our group also has access to, and this really helps eliminate any downtime trying to contact the owner of the document to make changes if we find a certain protocol can be edited to better suit our research. This is one of our research folders. And this is just where we kind of keep all of our protocols, the written ones, stored. New to our research group this semester is Flipgrid, and most of us have Flipgrid as an app on our phone, and it's where we store our video protocols that we have began making this semester. We have found that this recording our protocols has really played into people who learn visually over just reading the written protocols. It's also actually really user-friendly, not only in accessing the videos, but in recording the videos. There's not really any editing that needs to take place. You can just record in clips, and then it uploads maybe a 30-minute experiment into 2 minutes or shorter. So this is what our Flipgrid page looks like. We also use several lab notebooks kind of scattered throughout the campus where we do a lot of our research and in the room where we keep our Aplysia in. We also have individual lab notebooks that every member of the group got when they first started their semester of research. In these, we kind of just keep our daily tasks and observations as we're going through. And in the shared notebooks, we record results, and it's kind of where we start the rough draft of our written protocols.
Some areas of focus for our presentation will be the improved care of the Aplysia californica, along with the standardization of protocols such as zone of inhibition, gel electrophoresis, Western gel and ink collection, and lastly, the possible utilization of these protocol videos to be used in future introductory biology level courses. In this presentation, we will discuss how we standardized these protocols, how we adjusted the protocols through the semester, and how this has enhanced our overall research. We chose to focus on these areas in order to produce more consistent and continued research from semester to semester. We have also found that having a set of routine and instructions allows for research students to contribute more successful results to research.
One of our focuses this semester was standardizing the care of the Aplysia. In the beginning of the semester, we created a Google Sheet where each research student signed up to take care of the Aplysia daily. The daily tasks included cleaning and feeding the Aplysia. We created a feeding protocol video, which we'll show now, that demonstrated on how much algae we should feed each day to the Aplysia.
Alright, I'm going to be showing you the appropriate amount of algae to feed to the Aplysia once daily. So you're going to get your algae, just a small amount. It's green, normally it would be red, so it's easier to see that you just need a small amount. So then you're going to take your algae, give it to one of the Aplysia, and you're going to repeat this for all of them.
These videos throughout the semester were really beneficial for new research students like myself to refer back to to properly feed and take care of the aplasia. Another thing that we created this semester was the care sheet and the daily checklist. The daily checklist and the care sheet were something we filled out each day after taking care of the aplasia. This made sure that we were cleaning the tanks, taking care of them properly, and we were checking for signs of life in the aplasia to see if we needed to improve the care in any way. The care of the aplasia proved to be very significant over the semester. It led to more humane care, longer lives for the aplasia, and it also led to better results within the ink. One of the things that we noticed was that the aplasia were living longer. In previous semesters, they had to make multiple orders for aplasia, but this semester we started out with 6 aplasia, which have lasted us through the whole semester. We even started with 2 from last semester because they improved their care in the previous semester. We also found out and can accredit the care to the life of the aplasia in the inks that are much better results. One thing that we noticed is that the inks have a better consistency and color and run in our tests a lot better.
Zone of inhibition studies have been imperative to our research this semester because we're overall testing the antimicrobial activity of the ink of the aplasia. We use these zone of inhibition studies almost every week. So it is important for all members of the group to understand and be able to access this protocol whenever they need it. Recently, we've made some changes to our protocol. We've began studying the spun down ink in the supernatant and pellet and using those to test the antimicrobial activity of each of those. We've also found that using a standardized amount of each sample to soak each disc has improved our results as well. Our lab notebooks have also been important, where we record the measurement of each inhibition zones, and that's just something we keep a separate notebook for. And then we also know that it's important for all members of our group to be measuring the inhibition zones in the same way. We measure from the edge of the disc to the least amount of inhibition, and that's what we record. If one person measures incorrectly, then our results are skewed. And this is what our written protocol looks like. We like to try to keep it concise, but also descriptive enough to follow.
The next standardized protocol that we will discuss is the gel electrophoresis. Some specifics that we have standardized for this protocol is the use of a 1.5% agarose gel, uploading exactly 30 microliters into each well, using a 1x running buffer, and running each gel at 100 to 120 millivolts for an allotted period of time. This allows for equal opportunity of each gel to yield consistent results. For a student wishing to properly perform a gel electrophoresis study, it would be useful to view either our Flipgrid video or the lab notebook.
The first step in performing a gel electrophoresis experiment is to load the gel into the electrophoresis box. You can find how to make the gel at a separate video. Very carefully slide the gel out of the casting and into the electrophoresis box. The next thing you should do is record which samples go in which wells, labeling them 1 through however many you have, so that you're able to correlate the results with the chart. Be sure to notch or remove the first little section in the top right corner to keep straight which way is up and which way is down, so that you know which well is 1. Be sure to set your mechanical pipette to 30 microliters, or whatever quantity you choose. Place the tip on the pipette and suck up the proper amount of microliters into the pipette. Make sure that you have used the proper sample for your first well. Carefully insert your sample into the well. Go slowly at this to make sure you have no bubbles or leakage out of the well. After filling all of your wells, pour your running buffer into the machine. You should start by filling each side. Be careful not to pour too quickly to mess up the gel, and allow the buffer to run over the gel and form a small layer on top. Place the lid on the machine, making sure to line up the black side with the black side and the red side with the red side so that it runs properly. Turn your machine on and set it to between 100 to 120. You can allow that to run for however long it takes for your specimen to start running through your gel. We're going to leave it for about 1 hour. One tip is to check for bubbles forming from the bottom up. That will help you to know whether you have it set correctly and have the proper buffer in it.
As you can see from the protocol video, students are able to independently and successfully perform a gel electrophoresis experiment. This consistency has been proven over the course of the semester from each gel electrophoresis trial. This consistency allows us to further investigate these results and to determine what they may indicate.
One of our newer pieces of our research this semester was the use of a Western gel. We began the semester by using the manufacturer protocol that came with the equipment, but over time we made advancements to our protocol to better fit our research. To make these changes, we began by putting them in our lab notebook that was shared amongst the group, and then we referred back to and put it into the Google Doc that was shared amongst each other. Throughout the semester, we also created a Flipgrid video, and then for the staining and the preparation of the gel. These led us to be able to see the protein ladders in our gels and to make improvements. It also taught us that we need to be meticulous on following our own protocols and making sure we follow them directly, because one time we accidentally used a 10x running buffer instead of a 1x, and it took some time to figure out why we did not yield any results. Although it was kind of frustrating, it was definitely a good lesson for us to learn to follow our own protocols and to revert back to our lab notebooks and our protocol videos. One of the most important pieces of our research is collecting the ink. Ink is used in each of our tests, as you'll find out in the presentations and the poster presentation next week. It is important to collect the ink when we need the ink, and each of the Aplysia are inked every other week on either a Tuesday or a Wednesday. We can show you the ink protocol that was established last semester and into this semester that has allowed us to retrieve the ink from the Aplysia when it is needed. So as you can see, we take the Aplysia out of the holding tank, and then we'll stimulate its antennae and flop it back and forth in our hands 10 times. We then will gently set it into the collection bin, where we'll put a small piece of instant ocean water onto the Aplysia and wait for the Aplysia to ink. Sometimes it can take a few runs through the protocol for the Aplysia to ink, but it typically yields a very good amount of ink from each Aplysia. As you can see, the Aplysia's ink is a dark purple, and it can either be very watery like this is in the beginning, but over time it can become more viscous throughout the ink. We use a small manual pipette to take the ink from the collection bin into the collection tube. As you can see, the Aplysia ink, a very viscous ink on the end, and it is a pretty good amount. The Aplysia can ink different amounts each time, and we can often collect multiple tubes. But one thing we are trying to do, standardize the rest of the semester, is to find a way to collect the ink a better method. By using the manual pipettes, we often have to use a large amount of water, which dilutes the inks and can change our results. So we're looking into possibly using a vacuum system or a larger manual pipette to remove the ink from the collection bin and into the tube. Once we do that, we'll put the the protocols into the Google Docs and update our Flipgrid. That way we can have a cohesive research into the next semester.
Throughout our research, we have learned just how important the basic lab skills are, whether it's something that actually pertains to our research or it's more maybe necessarily not one of the experiments that we use, but we have began recording these basic lab procedures and uploading them to Moodle. We hope that introductory biology courses can utilize these when they teach, maybe even starting next fall. Here is our video on how to use a micropipette.
All right, today we're going to go over how to properly use a micropipette. So first you set the amount of microliters. So this is a 20 to 200 microliter pipette. So we're going to set it to 25 microliters. All right, and then the key is, so you're going to, when you press down, there's a stop. You're gonna feel resistance. So you always wanna stop at that first resistance and not go past it. If you stop here, you're going to pull up 25 microliters. If you go all the way, you're going to pull up 50 or more microliters. So we're going to demonstrate that. So in our first tube, we're just using some loading dye as an example here. So if I stop at the first stop, and then I put my tip in, pull up 25 microliters, and then I put that into this tube. Now for my second tube, I'm going to go past the first stop, go all the way down, pull it up, and then you're going to see that I get significantly more. So this was our first tube where we stopped at the first stop, and then this is our second tube where we went past. So again, when you're using your micropipette, always stop at the first stop, and that's going to give you the quantity that you actually set the micropipetter for.
Some future goals for our research group is to continue consistently updating the written and video protocols as our research grows. We are currently working on using the IR machine in the chemistry department, and we hope to come up with a good written protocol that works for our research and then eventually video that protocol and upload it to our Flipgrid so future members will be able to use that. We also hope to continue videoing the basic lab procedures and get those uploaded to either YouTube or another public video sharing platform where not only students at Quincy University can use them, but other students, maybe even in a high school, could learn from them as well.
Some acknowledgements that we would like to make as we conclude our presentation is first all members of our research lab group, along with Mr. Travis Yates, who first introduced Dr. Hale to Flipgrid and has allowed us to much better organize our research. And lastly, Dr. Michelle Combs, who helped with establishing the use of the Western gel in our research course.
I definitely think right now we're focusing on more humane and ethical research within our own group, but we're looking at possibly like writing papers on our protocol for the to ink our Aplysia. And so I think that we can include how we improved our care to make more humane care and longer lasting Aplysia in our research in that protocol, so we can maybe help other universities who are doing similar activities. They can use that to get better ink, because we did get better ink from our care. So I think that those two would go hand in hand in that paper that we want to publish.
So Flipgrid was introduced to our mentor, Dr. Hale, by Mr. Travis Yates, who is also a professor here at Quincy University. So last semester in research, we didn't have a set platform where we could kind of share with other members of the research group what exactly we were doing and what results we were getting. So if you had missed, say, like a day or a few days of research, it would be really easy to kind of get off track or feel lost, and then you don't have the results or the exact protocol, especially as we adjust the protocols through the semester. So at the beginning of the semester, Dr. Hale set up Flipgrid for us to use so that each research member could stay updated as the protocols changed. And we've seen a drastic change in the successful results that we've produced, and especially in the longevity of the aplasia.
Right now, I would say one of the biggest challenges is just trying to get a consistent way of actually like sucking up the ink from each aplasia, just because each aplasia's ink is so different. Sometimes we have like the older aplasia tend to ink more watery and like less, and it's hard when you're inking because they're on like a set schedule. If you ink an older aplasia with a newer aplasia, and that newer aplasia gives you really viscous, thick, dark purple ink, and then you have to use a lot of water to dilute it and get it to actually like suck up, and then testing those almost like against each other. So I think that's probably one of the biggest struggles that we're trying to work out now, is how to make that more consistent. Yeah, or like even just a bigger pipette, because it almost comes in like chunks of viscous, and they're so small, the little pipettes, that it doesn't quite get everything, and that's why we need to dilute it.